public welfare systems and society. Altering the macroeconomy, microeconomy in many societies. Actually, this can bring health problems such as the Alzheimer's disease cases. The incidence can double every 5 years after age 65. And almost of 50% of new cancer cases are diagnosed in patients aged 65 years and older as well. Then all age-related disease can be a big problem of health system. In this context, our symposium brings some hot points about this issue, bringing our speakers will bring different approaches about phytochemicals, about modulation on microbiome, and effects of exercise. As well, the oral communications, the selected oral communications can bring new perspectives about phytochemicals and pharmacological approaches. Then let me introduce our speakers. It's my great honor and pleasure to introduce Professor José Vinha and Dr. Heck Moraes. Professor José Vinha obtained his degree in medicine in 1976 and PhD in medicine in 1968. Noteworthy, doing research with Professor Hans Krebs, who identified the Krebs cycle in Oxford. Professor Vinha is full professor and chair of the Department of Physiology in University of Valencia, Spain. Last 30 years, Professor Vinha has studied different aspects of aging, including exercise and nutritional impacts on healthy aging and Alzheimer's disease. He has acquired great expertise in this field, what is important for our topic. Professor Vinha has received numerous awards and honors. The last one, the prize of Best Research Translatable to Society of the University of Valencia. Furthermore, he has been awarded with 2 Doctor Honoris Causa by Buenos Aires University and Rennes University in France. Dr. Vinha was the president of the Society for Free Radical Research International. Thank you so much, Professor. And let me to introduce Dr. Heck Moraes to be fluid in our presentations. Dr. Heck Moraes received her master's degrees from Federal University of Santa Catarina in Brazil, and had her doctoral degree at University College Cork in Ireland. In 2018, Dr. Heck Moraes joined the Masmanian Lab in the California Institute of Technology, Caltech, in Pasadena, California, as a postdoctoral fellow aiming for improving the understanding of gut-brain signaling in neurodegenerative disease, such as Parkinson's disease, using transcriptomics, immune and molecular biology techniques combined with pharmacology approaches. Understanding the relationship between gut bacteria and disease, that can help to design novel therapies that manipulate gut bacteria for the treatment of Parkinson's disease. It is an honor for us to have you both. Our first speaker, Professor Vinha. Welcome to our room.
Thank you. Thank you.
Thank you, Professor.
I am ready to start when you want. Shall I start now?
No. Yes, I can see. Thank you, Professor. Welcome.
Thank you. Okay, I need to have the screen of the organizers so that I can start my presentation.
Professor,
Yeah.
can't you see it? I'm already sharing and the participants are already seeing the screen. I
Okay.
think your Zoom is on window mode.
All right, so do we put the next slide? Next slide.
Just click twice on the Zoom.
Twice?
Twice on the Zoom slide, on the Zoom, so you can see your slides too.
Yeah, I can see it now, and I click twice.
Okay, perfect then. Let's proceed.
Okay, so next slide, please. Hello? Next slide. Hello? Victor?
Professor, aren't you seeing it? We are on the human and economic trauma slide.
Oh, no, I'm not there. All right. I can't, therefore, I'm not seeing your screen, Victor. I'm just seeing something that I believe to be my own screen.
Aren't you seeing the Zoom?
All right, now I have it. Okay, okay. So I now have it. All right. Good afternoon. Can you hear me all right, Victor? Yes?
Yes, we can hear you. Are you seeing the presentation now?
I see the presentation now. So the first thing I want to tell you to the audience is that the numbers are tremendous. In 2015, there were almost 50 million people with Alzheimer's dementia, and in 2050, it is expected to be 130 million. So this is 3 times. So this is a huge human drama. Now the economic was calculated to go from $600 billion to $800 billion per year. And this was in 5 years, an increase of 35%. All these data are from the prestigious Alzheimer's research in the United Kingdom. Next. And of course, Alzheimer's is clearly an age-related disease. Next. That's why a team like mine, we work on healthy aging. The name of the team is Fresh Age, and we attack it from different problems, from different fronts. And I'm going to discuss with you some results on a phytoestrogen approach to the treatment of Alzheimer's. Next. What we want, and I want the audience to know, is that you cannot treat the patient next when the brain is as in the third image, when the uptake of glucose is very poor. The uptake, if you see the presentation as it goes from the left to right, the left is the normal, then on early Alzheimer's to next one, and you see that there's a lack of, a lack of glucose uptake in the living brain, and then a late Alzheimer's, and that's the one you're rightly pointing at, and then you see how similar the late Alzheimer's is to the child. Child is to your right. The only thing is that the child is going to develop and the late Alzheimer's cannot develop. So first of all, you cannot expect a late Alzheimer's patient to behave as an adult, but rather as a child. And you cannot have the transition to the left, i.e. from the late Alzheimer's to normal. Next slide. What we have to aim at is to try and delay the onset of Alzheimer's, i.e. when we start the minimal cognitive impairment, we want to stop there. I will discuss with you how did we ever come to work with the genistein in Alzheimer's and the multimodal mechanisms of this phytoestrogen, and the clinical trial we have just completed and that will be published next week. Next slide. So it all started with this paper in the New England Journal. This was a review paper. Next, on a drug that was bexarotene that binds to the RXR. This is the receptor where the arrow points. And next slide. These people published in Science in 2012, this is, as you realized, 10 years ago, that this drug that binds to RXR, so the receptor, and activates the production of ApoE, reverses the deficits in mouse models. Next slide. And you see how the number of plaques falls down after treatment with bexarotene. This was a big success, and it was published, I just showed you, in Science, and it was reviewed in the New England Journal of Medicine. This is to give you a glimpse of how important this discovery was. Next slide. Very importantly, using the APP-PS1. This is a double transgenic model of Alzheimer's that we have used also, so same model. These people in Cleveland with bexarotene realized that it improves cognition, as shown in these slides. Next slide. So we thought that instead of activating the dimer, next slide, with bexarotene, we could activate the dimer next with, so the other side of the dimer, PPAR gamma, with genistein. And genistein is, okay, next slide. So if you compare with bexarotene, genistein is a phytoestrogen that is non-carcinogenic, all right? There's no risk of cancer. And next slide, bexarotene has, next, is antineoplastic, has side effects, and is expensive. Genistein is a food component, very few side effects, no carcinogenic, and is inexpensive. So we thought that we could use the strategy of determining the effect on cognition, next. Then seeing the histology and seeing the plaques, next. Then analyzing the molecular effects on A-beta, next. And finally, if we were successful with animals, try a clinical trial. Next slide. The first one, I will not go through all the animal experiments because I want to discuss with you more the human experiments. But this is a maze that can be easy or complicated, and is called the Webb. Next slide. Is the Hebb-Williams maze, and see in always the same code. The black is the white, the wild type, and then see when the animals are ill, the red, they take much more time to be successful in the maze. And when the animals are treated either with bexarotene, yellow, or with genistein, blue, the effects are very significant. In fact, we could say that from the viewpoint of the animal effects on this model, the bexarotene or genistein cure the model. Be aware that I'm not saying cure humans with disease, it cures the model, but it gave us hope that we could do something else. Next slide, please. And as you realize, as you probably know, the Alzheimer's patient lose odor, the capacity to smell. And you see the wild type in the black and red, the big loss in odor habituation, and how this is significantly improved with either genistein or bexarotene. So also with odor. Next slide. And then we went to the pathology. Of course, not myself, but from the biochemistry, but the people in pathology, and I will recognize them later on. And you see the arrows with the blue areas, those are plaques, and see how they are much fewer in the genistein. So they compare A with B or with C. So what we were doing was in fact confirming the American results, but also finding that genistein was as good as, if not better than, bexarotene. Next slide, please. This was now molecular. We determined by ELISA, by the molecular means, the amount of amyloid beta in brain. By the way, the mice don't have any amyloid. No amyloid in mice, so wild type is not here. But the disease animal, red, have a significant amount of amyloid beta in the brain that was significantly lowered by either genistein blue or bexarotene yellow. And it's both the 1 to 40 and 1 to 42 peptides. So genistein lowers amyloid beta as determined by histology or by biochemistry. Next slide, please. But it also lowers inflammation, and this is critical. This is determined by histology with a IBA, and you see the more yellow, the more inflammation. In the E panel, you see much more inflammation than the F panel with genistein or the G panel with bexarotene. So what you see is that genistein not only acts as an anti-amyloid, and this is important because it's special for genistein, but also as anti-inflammatory. Next slide, please. Now we did experimental PET and with fluorobetapir. This was a marker of amyloid beta, and you see in the picture, left picture is the diseased animal, right picture is the animal with a treatment with genistein, and see how it lowers. In fact, what we were doing with PET was simply confirming the experiments that we had done with histology. But of course, in the human, you cannot do the histology. So here, that was for the animals, and you see the mouse there, and how the genistein levels are much lower when the animal has been treated with this soya component, genistein. Next slide. And then we found the mechanism. The mechanism, to make a long story short, is genistein activates the production of ApoE and panels A and B, and that is mediated by PPAR gamma, because when we use an inhibitor of PPAR gamma, the effect is abolished, panel C. So it's a PPAR gamma mediated activation of ApoE production. That's the mechanism of the genistein effect. It's not just a food component. No. It's a special molecule, chemically defined, with a mechanism. Next slide, please. And in fact, we had been working on this for a long time. So from in 2006, we described that genistein is an antioxidant because it activates antioxidant genes of PANA. Then we also realized that genistein could inhibit the activation of P38, which is a marker of oxidative stress. Next slide. And it is antioxidant. So all these effects put together in the context of Alzheimer's disease models gave us, next slide, the idea that genistein exerts a general neuroprotective effect by activating ApoE production, lowering inflammation, and protecting against oxidative stress. So this is very important. It is a multimodal, and this is unique for genistein. Next slide, please. So we published it. And so what in 2016? And so what? Genistein cures Alzheimer's in a mouse. Be happy if you are a mouse. Of course, we needed to go, next slide, to a clinical trial. And this is the last part of my lecture. Next slide, please. A glimpse of what could happen came from analyzing the incidence of the death rate per 100,000 people. In Japan, 2.5, next, and in the United States, 24. So the rate of deaths in the United States and in general in Western Europe too, not so much in South America, but some too, is in fact tenfold that of the country where they eat more soya, so i.e. genistein component. This is no proof at all, it was simply an association. To get a proof, next slide, we decided to start a clinical trial. And we started it in 2013, and it took me a long time to have funds to cover this study, because genistein is not expensive, so industry are not really interested in promoting something that is so inexpensive. But I finally got funds from the Spanish government. Next slide. And the results will be published. This is the first time I say this in public. Will be published probably next week in Alzheimer's Research and Therapy, which is one of the best Alzheimer's specialized periodicals. The patients were receiving genistein or placebo orally for 12 months. This is 1 year. Next, please. The primary aim of the trial was amyloid beta deposition. Why? Because we thought, oh, it's not going to work in terms of cognition. So the secondary aims were the cognitive test. Next slide. What we saw is that genistein prevents amyloid deposition in the anterior cingulate gyrus in humans. Next slide. Okay, this is a very important area. Not in the rest, at least we have not seen changes in the rest of the brain, but the cingulate cortex, it plays a unique role in cognition and emotion. So at least we've seen that the amyloid deposition is lower in a critical area of the brain. Next slide, please. So then we came for the cognitive tests. Next slide, please. And now let me stop here for a second. I wish I was right in front of you and you could see me. We have tested 12 different tests, and in all of them, we've seen an improvement with genistein. But we've only found statistically significance in 2. So I'll go past each one of them, but you're of course free to ask me any question you want regarding this. So the mini mental, which is a very much used, see always the blue is control and the yellow is genistein. Genistein patients lose less than controls. Next. Okay, the croc drawing test, the same. Again, the blue is bigger than the yellow. Next. This is in this case, we reach significance. The TAVEC. TAVEC is a test where you they present you with 16 words for 5 times to evaluate the different memory and learning processes. And you see how the patients treated with placebo. By the way, the clinical trial was a double-blind, placebo-controlled, bicentric study. And I will give you the weakness later on. So the TAVEC is significantly better in patients treated. Next slide. And the delayed TAVEC as well. Next slide. Now, the Barcelona is for semantic verbal fluency. This, of course, has to be adapted for each country or each language. And this, of course, was performed in Spanish. And see that patients who were treated with genistein, the yellow bar goes up. In fact, they did not lose this fluency, whereas those treated with placebo did. Next slide. This is when people have to repeat the words after a few minutes, and you see again that the blue is much bigger than the yellow. They improve on this too. Next slide, please. And this is patients have to reproduce a complex geometric drawing. Again, the yellow is much smaller. The people who were treated with genistein lost far less than those treated with placebo. Next. And this is in percentage. And again, in this case, we reach significance. So we have already 2 tests that are significantly different. Next slide, please. And this is another way of the same test. Next slide, please. And this is the same. No, not quite. Go to the one before. This is the same, but in percentage. And again, we have significance. So I've gone past quite quickly because of the constraint of time. 12 different tests, out of which 2 were significant. And in all the rest, no one exception, there was a tendency. So we were quite satisfied, but then we thought, oh, what a pity that we cannot put together the ones that are not significant. Next slide. Then we came across this critical paper last year in the New England Journal of Medicine, where the strength of this study with an monoclonal antibody anti-amyloid beta. So it's a specific for amyloid beta. Remember that genistein is multimodal, anti-amyloid, etc. So the strength is that the patients are 257 because they have better funding. And these very prominent researchers did a baseline, if you see in the second, where it says worse and better, the change from baseline and integrated Alzheimer's disease research score. So what these people do is put together all their tests, and then they realized that the placebo lost 10% and the antibody lost only 6%, and this was significantly different. The only shortcoming was, apart from the economic one, it was twofold. One, that you have to inject the person every week, and second, that they have amyloid-related cerebral edema. Cerebral edema causes tremendous headaches. So that was the problem. Next slide. By comparison with the genistein, and the weakness is that the previous study had 200 patients. We only have 25, 13 and 12. Okay. But even with those, we have the TAVEC and the RAY, where the 2 tests were on their own, they were significantly better. And we did an integrated score, like inspiring what we had seen in the American study, and we saw that the placebo lost 55.8, whereas the genistein lost 14, and the difference was significant. So when it is integrated, we see that genistein has an effect in delaying the transition to dementia from minimal cognitive impairment. Next slide, please. I'm finishing. This has huge cost. Alzheimer's is a tremendous disease. Next slide. And very importantly, if we delay the onset of Alzheimer's by 5 years, the number of patients will be halved because people die of other things, but they don't die demented, okay? So delaying, as we're hoping with genistein, can have a very significant effect on well-being and on the economy of the whole world. Next slide, please. And of course, there are plenty of top pharmaceutical companies trying different monoclonal antibodies. Essentially, it's always monoclonal antibodies, and these have the problem to begin with that they have to be injected. All right. Always the indication is from prodromal to mild AD, just as we did with genistein. Next slide, please. And this is very important. We have to start to treat the patients as soon as they are either in preclinical or in minimal cognitive impairment state, before they show any dementia. And we can delay the transition to the dementia, hopefully, and in our hands it works. And as I said, this is the first time ever I said this in published with the treatment with genistein I've just described to you. Next slide, please. This was the front page, and I'm finishing, of Scientific American. A success in the fight against Alzheimer's. This was essentially with changes in lifestyle. Next slide, please. Treating, this is Scientific American again, 2022, so this is this year. Treating Alzheimer's before it takes hold. The major idea I have, apart from letting you know about genistein, is that we have hope of doing something with Alzheimer's. 10 years ago, I would have said there's nothing we can do. Today, and then I'm describing one treatment, this is with this phytoestrogen, we have hope we can delay the transition to dementia. Next slide, please. So epilogue, Alzheimer's disease poses a huge personal and economic problem, but, next. The advances in molecular biology, next. The new diagnostic tools I haven't had time to describe. The new drugs, like the one I told you today, genistein, and there's reasonable evidence of success in the treatment of human AD, as I've shown you in our clinical trial. Next. So there's room for hope in a new era in the treatment and the management of the disease. I'm not saying that genistein is going to be the treatment. It may be one of the treatments, and what we're hoping now is repeating the pilot clinical trial I have described to you as a full size with 250 patients, and we're trying to find funds for this. Next slide, please. And I want to thank the collaborators. Next. We have specialists in cognition, in animal cognition, and in histology, and in molecular biology from my own lab, and then for the clinical trials. And I have to thank all these colleagues from very different areas that have helped us to propose this treatment to delay the onset of Alzheimer's. And finally, next, and these are my research group, and they're very proficient, and they help me, and we're all fighting to try and delay the ravages of old age and promoting fresh age, as in this case, we're delaying Alzheimer's disease dementia. Thank you very much for your attention.
Thank you, Professor. Congrats for your results and your remarkable work. We can see that we can hope. I would like to ask you, the ApoE, did you measure in which samples?
We met ApoE, we determined it in vitro in culture of astrocytes. Astrocytes are the ones that make ApoE. So we culture human astrocytes, and we determine the production in the different concentration of genistein. And when we had one that was working and increasing the production, then we put an inhibitor of PPAR gamma, and it didn't work. So it's mediated by PPAR gamma. But remember, there are other effects like anti-inflammatory and antioxidants that work for different mechanisms. And this is the beauty of this molecule. I'm not selling anything. I'm just telling you the facts.
So exciting. Yes. The audience, someone would like to ask you something? Please. You can send in the meeting chat.
In the chat. I don't see anything. I have opened up the chat.
No, for a while. There isn't. Great talk, Professor. Thank you very much.
Thank you. It's been an honor and a pleasure.
Next time in Brazil, in physical meeting, hugs. Thank you very much. Congrats.
Thank you.
Hi. Can you hear me well? Can you hear me well? Hi, can you hear me well?
Yes, very well.
Yes, we can, but
Yeah, nobody's replied, so
it's not on presentation mode yet, but we can
it's
hear
not
you.
a presentation. Okay.
Don't
No?
worry. Perfect.
Okay. Sounds good. Yeah, so thank you so much for you and Nara to welcome me to present my postdoc data here today in the lab of Professor Sarkis Nizmanian. And it's an honor to present in a session with such great speakers, and it's quite intimidating as well. But today I'm going to present also another view of neurogenetic disorders and the relationships of our gut microbiome and brain. And my presentation is a little bit more focused on the effects of gut microbiome modulation of oxidative stress and mitochondria, as I'm gonna start introducing it now. So when everybody thinks about mitochondria, we kind of remember that they are the powerhouse of the cells, like in charge of great part of our cells' ATP production. But also mitochondria plays an important role on oxidative stress modulation, inflammation, regulates cell cycle and cell death. It also regulates our metabolism as it regulates the availability and usage of metabolites. And because of these functions on regulating the availability of a lot of biochemical and with neuroactive components, it can also modulate neurotransmissions and neuromodulation. As for example, mitochondria can regulate availability of glutamate, which is the main excitatory neurotransmitter of the cell. And the relationships between mitochondria and bacteria are quite ancient, and I think people are familiar with the endosymbiotic theory, which is one of the theories to explain the origin of eukaryotic cell as we understand nowadays, and also one of the theories to understand how mitochondria evolves. there are hypotheses that mitochondria may have evolved by the engulfment of an aerobic prokaryote by another organism. There are like polemics on which organism was the one that probably engulfed these other aerobic prokaryotes, but that endosymbiotic relationship makes us originated the eukaryotic cells as we understand. And there are remainings of these relationships between ancient relationships between mitochondria and bacteria, as here very well described in the context of infection, where a lot of pathogenic bacteria, they can release factors that are transported to the inner side of mitochondria and trigger apoptotic events during infection. So this relationship has a long-standing dynamics, and which I call the contemporary paradigm is that we are trying to understand the relationship between a gut microbiome and a brain mitochondria, which like can seem really further away, but I will try to explain the logic of it during my talk. So recently, we are becoming aware of the relationship between our gut microorganisms and the brain, and how this can regulate behavior and brain function in health and disease. And it has been described that the brain can regulate, can be linked to the abundance of different microbes, and also the gut bacteria can send signals to the brain through direct pathways, which involves the vagus nerve and enteric nervous system connection, but as well as through the secretion of cytokines into the periphery that can penetrate the brain and trigger brain inflammation, and also activates the innate immune cells in the brain microglium. But also the gut microbes can regulate the production and secretion of biochemical molecules that can be metabolites that can have neuroactivity. And the gut microbiome changes have been implicated to several central nervous system conditions, such as Alzheimer's disease, multiple sclerosis, Parkinson's disease, autism spectrum, depression, stress, and a number of diseases every year have been implicated to gut. And the rationale to think that there is an interaction between gut microbiome and mitochondria also comes from the fact that gut bacteria can modulate the production of several metabolites, such as tryptophan, fatty acids, branching amino acids, and other amino acids, and pyruvate, TMO, that they can trigger mitochondrial events, either by having receptors infecting mitochondria or by indirect pathways. And as a topic of this session, it also involves aging. So during aging, there's a lot of mitochondrial stress, including the increase in oxidative species production, and because our defense mechanism against oxidative stress is impaired, goes in impairment during aging. But there are also other changes in mitochondria that are involved during aging, such as mitochondrial genomic instability, inflammation, as also a result of mitochondrial genomic instability, so the release of mitochondrial DNA into the extracellular compartment. There are metabolic shifts, so there are changes in the use of the substrates. for production of energy. And those events can accumulate and regulate cellular senescence and apoptosis. One of the aging diseases that are associated with mitochondrial deficits is Parkinson's disease. So here, just showing one of the first evidence that Parkinson's disease, individuals with Parkinson's disease, they have a deficit in mitochondrial complex 1. Here is in post-mortem brains, but also this fact has been replicated many, many times, both in humans and also researchers have been using that to model Parkinson's disease. So exposures of neurotoxins that can bind and come to complex 1 and trigger complex 1 deficiency, as well as studying genes that are associated to mitochondria, and they can also affect, and they are also linked to Parkinson's disease. So here showing it's more, so Parkinson's disease, we don't have a cause established for Parkinson's disease, but it's believed that there are environmental factors and host genetic factors that may be regulating Parkinson's disease. And it's interesting to see that a lot of those factors that are associated with Parkinson's disease, they are implicated to mitochondria. Here, as examples, host genetic factors that may increase the odds of developing PD, such as alterations in expression of parkin, ATP13A2, DJ1, BINC1, LARC2, alpha-synuclein. Those are genes that are related to increased odds of developing Parkinson's disease. Also, on the other side, external side, there are environmental factors that may be linked to the increase of PD, such as exposure to toxins, paraquat, rotenone, that can inhibit complex and mitochondrial complex, respiratory complex. Aging is, as we see in the other talk, that it's a really important risk factor for neurodegeneration. Different diets have been associated with increased or decreased odds of developing Parkinson's disease, and they also are linked somewhat with mitochondria. So we're interested to our lab is interested to understand what's the role of the gut microbiome, and that lies in the interface of the environment and the host to modulate Parkinson's disease. So Parkinson's disease, our understanding of PD have changed over time. So before we used to characterize Parkinson's disease as just a motor behavior syndrome, but the more recently we have been understanding that Parkinson's disease actually can affect several physiological systems and can implicate a lot of non-behavior, non-motor symptoms, such as cognitive deficits, but also sleep disorders, loss of sense of smell, and gastrointestinal issues, which seems to be highly prevalent in PD patients. So around 80% of the patients that are diagnosed with PD, they have gastrointestinal issues. And sometimes these gastrointestinal issues starts a lot earlier before someone gets diagnosed with PD. Some key neurological signs of PD, pretty well established, is the neurodegeneration of nucleostriatal neurons and the aggregation of the protein alpha-synuclein. So, and it's interesting to see that, in fact, this very important hallmark of PD, the aggregation and phosphorylation of alpha-synuclein can be observed outside of the CNS. So there are some reports that show that alpha-synuclein can be observed outside in the enteric nervous system as well as in the sympathetic nervous system. So leading to us to hypothesize that perhaps there is a gut-to-brain connection or a brain-to-gut connection in the development of PD, where maybe alpha-synuclein pathology can perhaps start earlier in the gut and being transferred to the brain. So we are interested to understand what's the role of microbiome also in alpha-synuclein pathology. In PD, there are changes in mitochondrial microbiome composition. Here in green, some taxa that's have a reduced abundance in PD patients. Here in blue, some that are increased in PD patients. And it's interesting to see that there are also functional implications to that those taxa that are altered in PD patients, they also regulate differential abundance of neurotransmitters and amino acids and several microbial linked metabolites. So that can have functional readouts, and we're still trying, it's 30 days for metabolomics research, and together with metagenomics to understand what the role, functional role of those changes. So this is the first study that developed in trying to understand the role of the gut microbiome in PD in our lab. So Team Samson has shown that when this animal model of PD that's based on the overexpression of alpha-synuclein, and these mice are re-derived to germ-free conditions, so they are born and raised without any microbes. They have a less pronounced pathology, so they behave better, their motor symptoms are better, and they also don't have as pronounced alpha-synuclein pathology in the striatum and substantia nigra, which are important regions for modeling PD. So it is showing that the gut microbiome, there are factors in the gut microbiome that may be leading to pathology in this animal model. And what I did was to look at the striatum and isolate the mitochondria from neuronal cells. And what we see is that also the alpha-synuclein protein, while it can accumulate in the mitochondria of the mice that are raised with microbiomes, which I call here ASO-SPF, in our ASO germ-free, in the lack of the microbiome, they don't have accumulation of alpha-synuclein in the mitochondria. So we want to understand if these would have functional implications. So with this isolated mitochondria, we ran the scores. So we simulated the mitochondria with substrates that can trigger complex 1 and complex 2 respiration. And what we see is that in the SPF-ASO, there is actually an increase of mitochondrial respiration, both for complex 1 and complex 2, in comparison to the germ-free counterparts. So that was quite surprising to me, because what I was expecting was the opposite, that there would be like a deficit in the SPF-ASO. But what appears to be happening here, which is we are just able to study a very early stage of the disease because of time constraints of research. So these mice have 4 months old. So it's a very early stage of pathology. So we believe that those are compensatory effects for some other damage that's happening in the cell. But here also really demonstrating that the microbiome drive respiration in the striatum. And there is also some confirmatory data when we did Western blot with this isolated mitochondria. We also see trends to increase in expression of proteins that are important for the formation and of complex 1 and complex 2, as well as we see trends of increase in ATP production in SOSPF in comparison to germ-free ASO. It also makes us think that the cell at this stage is needing more energy for some reasons, such as maintenance of neuronal activity. We also perform brain transcriptomic studies. We did RNA-seq, bulk RNA-seq. So here it's the overall cellular transcriptomic. And what we see is that in the differential expressed genes between our wild type SPF group and ASO SPF, we see an enrichment of genes that regulate synaptic function in the wild type SPF in comparison to ASO SPF. While in the ASO SPF group, we see an enrichment of genes that regulate energy metabolism. So really like confirming our results from the seahorse that maybe there are some more activity in the ASO SPF groups. Also, when we compare our model in the SPF and in the germ-free forms, we see the same thing, that the ASO SPF has an enrichment of ATP and mitochondrial and cellular respiration associated genes in comparison to germ-free counterparts. We also look at the proteome, and here we enrich fractions for mitochondrial proteins. And what was interesting is that we see that in the ASO germ-free, which seems to be have a better motor performance and like kind of normal respiration, we see that there is enrichment for proteins that are associated with oxidative stress. So it seems that if alpha-synuclein is triggering oxidative stress, this group seems to be responding with increased expression of antioxidant proteins in comparison with SPF. So that is probably, so that's our probably what may be happening, but also the hypothesis we have been working now, which is based on those mice that have alpha-synuclein overexpression in the presence of, in the absence of mitochondria, of microbiota signals, they can respond different to oxidative stress. And this may be linked to alterations in mitochondrial respiration, as well as altered behavior performance. And in fact, we observed that in the ASO SPF group, there is an increase of oxidative stress in the striatum as measured by total oxidative peroxide concentrations. And this in comparison to the germ-free mice and also to mice that receive an antioxidant in their diet. And when we give this antioxidant their diet and we test for their motor function, we see that the ASO mice, they have a much better motor performance, so we're able to ameliorate this phenotype. And at the moment, we are trying to understand better what's happening with specifically neural mitochondria. So looking at the synapses and looking at those, the mitochondria that, and especially in presynaptic neurons, that what is happening with them in terms of function, but also morphology. Here, an image from Mark Ladinsky, where we just show that perhaps they have different morphology in comparison to SOSPF, but those are still a beginning of our analysis. And we are also isolating these mitochondria from presynaptic terminals and seeing their activity. And we are also interested in understanding what signals from the gut microbiome could be actually linked to mitochondria in the brain. And this is probably a question to be addressed in future research, but we are starting to do that. And some other groups are also trying to investigate similar questions on how gut microbial metabolites can alter mitochondrial function. Here an example in studies from the group of Richard Fry and Sandra Cardozo that are looking at the effects of also propionic acid or branched amino acids to modulate mitochondria in brain cells, as well as here a recent study by Mark Briggs that showed this interaction involving microglial cells. Just very recent, was published last year. It shows that microbial-derived acetates can change metabolic fitness in microglial cells, depending on the presence or the absence of the gut microbiome, with implications to Alzheimer's disease animal models. So we are also performing a large study on metabolomics, where we did metabolomics with different parts of the brain and the gut of this animal model of Parkinson's disease, trying to find molecules that correlate between the gut and the brain. Here, just an example to show illustrate on that. The most significant molecules that we found, they are lipids, amino acids that are differentially expressed between germ-free and SPF. So we're still looking at the potential molecular signals. So I want to thank everyone that has been helping in the study. Of course, Professor Sarkis Mazminian, that we have a great lab, great environment, and helps with resources and mentorship. And Neela Freeman, Joseph Boktor, that starts as technicians. Now they're going to grad school, and just Joe stay in the lab, and Terrence also a lab tech in our lab. Fan Jeff and has been helping with bioinformatics, and Mark with EM studies, and the other team that has been helping a lot of outside Caltech that has been helping me a lot with metabolomics and metabolism studies, including Rima, Sia, Joanne, Christine, and Lindsay. So thank you very much. Yes, we have some questions.
Thank you, Lydia. Great talk. This is an exciting topic. I think so interesting, the relationship, the symbiotic relationship. This link of endosymbiotic hypothesis is very interesting. Very, very interesting. O pulo do gato. Very interesting. I was wondering about the short chain fatty acids, but you tell about acetate. Do you know anything about butyrate?
No, but I suspect that it's very related to metabolism, right? And so I suspect there's some effects. I haven't explored that yet. I did treat the mice with fiber diet, high fiber diet that can increase the availability of acetate, but not butyrate. So I haven't looked at iterated yet. But yeah, that's something that I'm very interested to do.
Another point that I was wondering is about the diversity of the microbiome. Most time we heard in general groups, and actually there are dozens, thousands of bacteria in our microbiome. Then, do you know about this? Which species or groups could be more important in prevent or treat the generation?
Yeah. So I think it's still early days, but we have been trying in our lab to research from some PhD students, like they're trying to treat mice with different probiotics to see if they can reverse some of these phenotypes. So really basing like our targets based on these systematic studies that have been. So seems like Archementia muscifera. So it's a microbe that have been really associated with metabolic disorders, but also we've seen an enrichment in animal models of PD. So and I think that has been replicated in more than one model. So that seems to be a good candidate, but it's hard to tell which one. It's still early days for this research, and this research needs to be replicated in several animal models and humans, right?
Great.
Yeah, that's interesting.
Thank you.
Thank you.
Professor